Good morning, everybody. Sorry I have not been super active or really active at all in checking in on how you're doing this week, but hopefully you are making progress on your group projects and on the muscle summarizer and understanding how the sarcomere works. So, what I want to do this morning is just talk about the kidneys and how they excrete waste and maintain water balance. Which is very important for them to do. The kidneys are a very important organ in our bodies that you might not know a ton about, but hopefully you will understand a little bit better their function in our bodies after we go through this lecture. It's pretty short, but all of the slides have a lot of content on them, so take your time as you're going through and feel free to go back or pause me as you're trying to understand the kidneys for yourself. Okay, so the first thing we want to start with is the fact that there are these things. All organisms are either osmoconformers or osmoregulators. And what that means is that you can either regulate your concentration of water on the inside of your body, or you can conform to the concentration of water and solute inside of your body based on your environment. So, if you remember from chemistry, and maybe some from biology as well, or freshman biology, osmolarity is the solute concentration of a solution. So, remember, solutes are the stuff that dissolve in the solvent. So, most of the time, the solvent is water. Solute could be salt, or sugar, or potassium, or chlorine, all of that. And when you have a lot of solute in a solution, you're hypertonic, and when you have less solute in a solution in comparison to the environment, you're hypotonic. And then if you have the equal amount of solute in your solvent as the environment that you're in, you would be isotonic. And remember that in osmosis, water always moves towards where there's more solute, almost like in an attempt to dilute it, because water is attracted to the solute. So in an isotonic environment where the inside of your body is equal to the outside, if you're an osmoconformer, you have no net loss or no net gain of water, because the water is going to be moving in and out between you and the environment, but because both sides have equal solute concentration, you shouldn't see any net gain or loss. But if you took like one of our red blood cells and you stuck it in a very hypertonic environment where there's more solute on the outside of the red blood cell than is on the inside of the red blood cell, we would see that the red blood cell would lose water to the outside environment. The opposite of that would be if you put a red blood cell in a very hypotonic environment. Because there's more solute inside of the red blood cell than outside of the red blood cell, then water from the outside environment would flow into the red blood cell, which for a time would be fine, but if there was a very big difference between the inside and outside environment, that would cause so much water to want to go into the red blood cell that could cause the red blood cell to lyse or burst. So if you think about that image with osmoconformers, they have the same osmolarity as their surroundings because they don't have a mechanism inside of their bodies that's going to help them to control their osmolarity. So they just end up being whatever osmolarity their environment is, or whatever osmolarity water is going to decide it is based on how water moves. It's definitely going to be a lower energy choice to be an osmoconformer because you're not going to put in energy to control your internal environment that way. But it's also going to be challenging for you to deal with changing environmental circumstances if you can't regulate your own osmolarity inside of your body, as the outside osmolarity might change. And then osmoregulators, like ourselves, we have adaptations like kidneys that can change our internal osmolarity from our surroundings. So that allows us to maintain homeostasis, where we can have an internal balance regardless of what the external environment is. We can also have that internal balance when our internal balance is attempting to be thrown off. So there might be times when we are producing more waste than we usually do, and we can still pump out that waste and keep our osmolarity the level that we perform best at. Even though there's an increase of waste, we can also cause our, for example, kidneys to make sure that we pump that waste out, so we don't have a increased concentration of waste, whereas osmoconformers would not be able to do that. So hopefully that makes good amount of sense, the difference between osmoconformers and osmoregulators. Osmoregulators, just have a better mechanism for controlling their internal environment if the external environment changes, so they don't have to deal with a sudden intake of water or a sudden loss of water as much. And then also, if their internal environment changes, they can try to bring their internal environment back to the baseline that they are most stable at. Okay, so like we said before about muscles, chapter 11 is called Animal Physiology, not Human Physiology. So we do focus on the human systems the most, but IB likes to insert some information about other organisms that do similar things like we do. So insects, they also osmoregulate, and they don't use kidneys, they use this structure called Malpighian tubules. So first thing that you need to understand is that whenever you metabolize or break down proteins, you end up making amino acids. And then when you metabolize the amino acids and kind of repurpose them, recycle them so we can do protein synthesis to build new proteins, that's going to lead to the buildup of nitrogenous waste. Basically just waste that contains nitrogen. And the other thing you want to understand is there are 3 major types of nitrogenous waste that animals make. They make ammonia, which is water soluble, but it's very toxic. So basically it means that ammonia dissolves in water. And then they also make urea, which is water soluble and less toxic. And then they also make uric acid, which is not water soluble and the least toxic. So we've got ammonia, urea, and uric acid. So the type of organisms that make ammonia as their nitrogenous waste are going to be organisms that live in water, and that allows the ammonia to flow out of them and then into the environment. So fish, they make ammonia waste. So when they break down proteins and amino acids, they make nitrogenous waste, they make ammonia. The ammonia is just kind of like peed out of them into the water around them. And then it's fine. They're getting that pretty toxic substance out of their bodies. For us, we make ammonia, but because we're not surrounded in water, the ammonia builds up inside of us. And we convert ammonia into urea, which is it's less toxic. And it's going to be the main component of our urine. So our urine is made of urea, which is a less toxic version of ammonia. And so we store our urine or our urea in our bladder, and then we excrete our urine, which contains the urea, a couple times a day. And that's fine because we have some water in there, which dilutes the urea, and we've converted the most of the toxic ammonia into urea, so we can carry it around for a while. If you weren't able to convert the ammonia into urea, it would cause some pretty big problems and build up in like your brain and cause brain issues. So it's good that we convert our ammonia into urea. And then there's organisms that don't have a lot of access to water, so they can't dilute their ammonia, they can't dilute their urine, so they take another step and they convert the urea into uric acid, which is solid or semi-solid. So birds make uric acid, so that's what mostly is, like if you find it on your car, that's uric acid. And bugs also make uric acid, so that's what we're going to take a look at. So Malpighian tubules connect to the digestive system, and what they do is they transport waste out from the body through the gut and then out the hindgut. And what we basically want to look at is this structure over here. It's kind of a weird picture. So you're imagining you're looking at the body of the insect here. It kind of looks like there's an insect inside of this like brown thing, but this is the back or hind part of the body of the insect. I think we'd say it's like the thorax of the insect. So imagine legs are coming out here, its head would be like right here, its like antenna would be here. Okay, so this is the GI system, which is this red part of the insect. And the GI system, or the digestive system of the insect, has these tubes coming off of it called Malpighian tubules. And basically, Malpighian tubules go into the body cavity of the insect. So here's the body cavity of the insect, and we've got these tubes kind of branching out from the gut. And what the tubes do is they collect waste that's being built up out here. So as these body parts of the insect start to digest and metabolize amino acids, more and more nitrogenous waste is being created. And so we might also have like excess water or excess salts and potassium. So these things will just kind of flow through active transport. So being pumped basically into these Malpighian tubules from these outside cells over here and over here. So water will be pumped in, and then sodium, potassium will be pumped in, and then uric acid, which is that waste, will be pumped in. Actually, I'm going to correct myself. Water is not going to be pumped in. Water is going to go through by osmosis. So if you pump sodium and pump potassium and pump uric acid, you're basically moving solute from out here to here. That causes these tubules to become hypertonic, causes the outside environment to become hypotonic, and water will follow solute wherever it's being pumped. So because the solute's being pumped into the Malpighian tubules, water is going to follow. So then you're basically pulling all of this excess from the outside cells into the Malpighian tubules, and they are going to empty themselves into the back part of the GI system of the insect. So it's the midgut in the kind of like the middle of its body, and the hindgut kind of closer to the back end of the insect. And all these waste products that have been collected are then going to just kind of flow through the GI tract, and then we might steal some of those ions back depending on if the insect needs some more sodium or needs some more potassium or needs some more water. It's called reabsorption. We do this also, which we'll talk about. And then what's left behind is really just waste. So you don't reabsorb the uric acid. Uric acid is trash, so you definitely want to dump the trash into the Malpighian tubules. They make their way into the GI tract, the midgut, then the hindgut. And then there's an opportunity to pull back things that your body does need, or the insect's body does need. So sometimes sodium, sometimes potassium, sometimes water. But the uric acid is going to stay because it is definitely a waste product. And as you're pulling back that water from kind of like the waste products, that's going to dry up the uric acid, and it's going to become kind of like a semi-solid waste or semi-solid paste, and that's what's excreted from the insect. So that's basically how insects osmoregulate, and the idea is basically that you're building up all these solutes, sodium, potassium, uric acid, and then water is going to also kind of dilute those things, and they're going to travel through your GI system, and then there's going to be a system for homeostasis in the insect that is going to know if we need to pull back more solute because we're running out of solute over here, or pull back more water, or we can leave those things in here and then excrete them. So these numbers will change. How much sodium, potassium, and water you pull back is going to change based on the internal environment of the insect and whether it needs to increase the solute that's in its body or decrease the solute that's in its body. If it needed to decrease the amount of solute that's in its body, then it would not reabsorb very much sodium and potassium. They would just stay in the hindgut, and with the uric acid, they'd be excreted. So it really depends on what the needs are for that one time. Okay, and I think that's pretty much it. I talk about Malpighian tubules every single time I teach IB Bio. It's just one of those things that people kind of get while we're looking at it, but then forget exactly what's happening when they look at it on their own. So I'm sure you'll come back to this slide at some point and try to understand Malpighian tubules, but that's all we really need to know for now. Okay, So humans don't have Malpighian tubules. We have kidneys, and kidneys are our osmoregulation organ. We have two of them. They are kind of on our backsides, lower backsides, and they maintain water and ion balance. So, kind of if you put like your hands maybe slightly above your hips, your fingers would, with your thumbs on the front side and your fingers on the back side, your fingers would kind of be where near your kidneys are. Okay, anyway, the kidneys filter blood, and when they filter blood, um, what's in the kidneys is called filtrate. And after the kidneys filter the blood and produce the filtrate, they make concentrated urine, which is basically just concentrated wastewater that contains urea. So basically, um, if you guys remember from the circulatory system, the aorta is going to pump blood out of the heart and around to the body. There's a bunch of top branches of the aorta, and then there's a descending branch of the aorta, which kind of goes down behind the heart. Then it kind of comes down your abdomen and abdomen, and then it's going to split left and right to form your renal artery. So each kidney has a renal artery, which is going to feed it. So blood's going to come through the renal artery into this kidney, and then the blood is going to be pulled out by nephrons in the kidney, which are like little filters in the kidney, kind of shown by like these little lines that we see. The nephrons in the kidney are going to filter the blood, basically means pull out what's in the blood, and then kind of decide what we want to keep, what we don't want to keep. And then all the stuff we don't want to keep is going to get emptied into the pelvis of the kidney, and then emptied into the ureter, which connects to the kidney. And the ureter is going to go to the bladder, which is going to store that waste. So anything that stays in the kidney is going to go into the ureter, into the bladder, out of your body. All the stuff that is in the blood that the kidney pulls out to kind of like take a look at, but then decide that we want to keep it, it's going to go back into the blood, and how it returns to circulation is through the renal vein. Oh, excuse me. Hey Google, turn off the timer. And sorry about that. And it's going to go through the renal vein and then go up the inferior vena cava and go back into your heart and back into circulation. So what you want to remember is if stuff stays in the blood, it goes back to your body. If stuff goes into your kidney and then stays in your kidney, it's going to go out of your body through the ureter, through the bladder. If stuff goes out of your blood into the kidney, then out of the kidney, it's going back into your circulation through your renal vein. We'll talk about that more, but it's important to know that if stuff's going to stay in your kidneys, it's going to make its way out of your body. Okay. Other things is the kidney, when you do a cross-section of it, it's got these three distinct parts to it. It's got the cortex, which is kind of like the perimeter of the kidney. It's kind of like when we talk about the cerebral cortex, which is the first layer of the brain, which is the kind of folded layer of the brain. We've got the medulla, which is going to be closer to the inside of the kidney. And then we've got the pelvis, which connects to the ureter. The cortex is lower osmolarity, which basically means the kidneys are kind of salty, and we'll talk about why they're salty. But the cortex is a salty place. The medulla is a really salty place. So it's hypertonic down here and less hypertonic here. So the deeper you go from the outside of the kidney to the inside of the kidney, it gets saltier and saltier. And we'll discuss why that's important, but it has to do with osmoregulation. The other thing we want to talk about is what's coming into the kidney and what's coming out of the blood. So what's coming into the kidney through the renal artery is going to be this stuff right here. And then what's going out of the blood and then into the kidney is going to be this stuff right here. And I really should add one more column which says what stays in the blood, but I'll edit that for another time. So basically, when we're talking about the renal artery, what's coming in, we call that plasma. And what's in the plasma is, for example, 151 moles of sodium, 110 moles of chloride, 5 moles of glucose, 5 moles of urea, and 740 milligrams of proteins. So that's what's coming in through this renal artery. So that's what's currently in your blood before it gets filtered by the kidney. Then what leaves the blood and goes into the kidney is going to be this stuff. So what we see is the blood had 151 moles of sodium. 144 moles of that sodium leave the blood and jump into the kidney to get filtered. So 7 moles of sodium stay in the blood and then they swing back out and they leave the kidney. And then we see 110 chloride ions were in the renal artery and 114 of them ended up in the filtrate. It's a little bit more than what was coming in because more is actually going to be actively pumped in from not the renal artery, but from the surrounding environment of the kidney. So just kind of ignore the fact that the numbers are a little bit higher, but just know that basically chlorine is coming in the blood from the renal artery, and pretty much all of the chlorine ends up in the kidney. So not a whole lot of it is coming back out through the renal vein. Then we see 5 glucose moles go into the renal, go in through the renal artery, and they all end up in the kidney. 5 for 5. Same with urea. The urea comes in through the renal artery, which is the waste, and all of it hops into the kidney. And then we see protein, 740 protein milligrams come in through the renal artery, but only 3.5 of them end up in the filtrate. So 760.5, sorry, that's not right, 736.5 end up going back out through the renal vein. So what you want to get from this last number here is that there are proteins in our blood, things like platelets, red blood cells, those are made out of proteins. Things like fibrinogen, which helps with the clotting cascade, prothrombin, which help with the clotting cascade. Those are all in our blood, but they do not go into the kidney. They actually swing right back out, and they return to circulation through the renal vein. So proteins do not end up in the kidney because the kidney is a fast track to the ureter and to the bladder, and we try not to throw our proteins out of our body because we made them and we probably need them for something. The only way that you really get rid of proteins in your body is if you break them down and you turn them into urea, and then the urea is then going to go to your kidneys and get into your kidneys and get thrown out by your kidneys. Otherwise, you kind of want to hold on to your proteins, and we'll talk about why you hold on to them. Well, we said why you hold on to them because they're important. You made them. They do a lot of things in your body, but also they're too big to fit into our kidneys, so we don't really toss out things that are large in our blood. We only toss out the small things that have been broken down. Okay, so we talked about what's going in is this plasma column. What is in the kidney is this filtrate column, and you could have a third column which says what comes out of the kidney. And what comes out of the kidney, which is going to be maybe cleaned plasma or filtered plasma, is going to be a lot of the salt is going to come back. We're going to keep a lot of that salt in our bodies. We like to have around 135 to 145 moles of sodium in our body. That's kind of like our nice balance level. The chlorine, we like to have about 98 to 100, I want to say 108, maybe 92 to 108, something like that. We like to keep that amount in our bodies for chlorine. Glucose, we pretty much are going to have the same amount of glucose come out that came in for the same reason with proteins. We'll talk about why the glucose ends up going from the plasma to the filtrate and then coming back into the blood, but you should have a 5 here if we're talking about what's in the renal vein, because we pull back all that glucose. And let's just talk about it now. We pull back all that glucose because food is hard to come by, and glucose turns into energy through cellular respiration. So if food is hard to come by and glucose turns into energy, we don't just want to throw it out, we want to save it. So the glucose that comes in through the renal artery and jumps into the kidneys is going to come back out through the renal vein. You don't really have sugar in your urine. You might have sugar in your urine if you are overflooded with sugar. So sometimes people who have diabetes, they would have sugar in their urine. And so if you guys might recall from diabetes, you can either have diabetes type 1 or diabetes type 2. Diabetes type 1 means you don't make insulin. And insulin is the hormone that tells your cells to absorb the sugar from your blood and then put it into your cells. So that decreases how much sugar is in your blood. People who have diabetes type 2, they do make insulin, but their bodies ignore the signal of insulin. So still, even if they make insulin, their cells are not absorbing the sugar from their blood into their internal cells. So whether you have diabetes type 1 or diabetes type 2, more sugar is hanging out in your blood and less sugar is being pulled into your cells. So what happens is that because there's so much sugar left in your blood, because your cells are not able to pull them in, because they're not responding to the insulin signal or they don't have an insulin signal, then there's lots of sugar in your blood. And when that blood goes to the kidneys to get filtered, because there's so much sugar in your blood, a lot of it does end up getting dumped out of your ureters and into your bladder and out of your body. So then you end up producing sweeter urine. And what they used to do before we had glucose tests or diabetes tests is the doctor would have to do like a little sampling of their patient's urine. And it would be sweet enough to taste the glucose if the person had diabetes. Anyway, fun fact. The urea comes through your renal artery, goes into your kidneys, that's the filtrate part, and all of the urea should pretty much stay. So it's 5 in, and 5 into the ureter. Pretty much nothing should come back through the renal vein. So if this is renal artery, this is inside the kidney, this is renal vein, it should be 5, 5, 0. Whereas glucose is 5, 5, 5. 5 goes in, 5 gets filtered, 5 comes back into your body. Urea, 5 goes in, 5 gets filtered, none goes back into your body, all of it goes out of your body. Your proteins, 740 go in, 3.5 get filtered, and then pretty much all of it is going to come back out. So that 3.5 is going to come back out and join the 740. So you shouldn't have proteins in your urine. Okay, so hopefully as I was going through that, it started to make more and more sense as to what's happening in the kidneys. So again, think about your plasma that's being brought by the renal artery, your filtrate, that's what the kidney is filtering, and then your urine would be what it's getting rid of, and then your filtered plasma is what's going through the renal vein, going back into your heart, back into circulation. And the kidney is going to do this multiple times a day, so it's going to do it every single time that the renal artery brings blood to it. Okay, so the next thing we're going to look at is a little bit more closely how the little nephrons that we mentioned that are inside of the kidney do this job. Okay, so the nephron is the functional unit of the kidney, and you guys have heard this term before, functional unit, like the sarcomere is the functional unit of the muscle, or the neuron is the functional unit of the brain. The nephron is the functional unit of the kidney, which just means the nephron does the work that the whole kidney does. So the kidney is able to osmoregulate because it is made of millions of nephrons, and each nephron helps to filter the blood, and that allows a whole kidney to be good at filtering the blood. There's a bunch of different parts to the nephron, which we want to talk about. So the nephron is this kind of goldish, brownish color, greenish, whatever you want to call it, that is made of these tubes. It's just one long tube, and then they all empty into the pelvis. So a bunch of these tubes in the kidney, in the cortex and medulla, empty into the pelvis. And then each nephron is going to be vascularized, which basically means that it has blood vessels. So you see these blue guys around the nephron, those are blood vessels. And that's important because we know the kidney filters blood. So how is it going to get the blood that it filters? It's going to need these blood vessels to give it that blood. And then how is it going to return stuff that it doesn't want to get rid of? It's got to have blood vessels around it so it can dump stuff back into the blood or pull stuff out of the blood. So that's important to know as well. So if we do the whole overview of what the nephron looks like, we'll start with the blood system. So we said the renal artery comes into the kidney, but stuff doesn't flow out of arteries and stuff doesn't flow out of veins. They have to get smaller so their walls are very thin so stuff can go out of those vessels. So the arteries are going to turn into arterioles, and the arterioles are then going to kind of turn into capillaries. And then capillaries merge into venules, which are very small veins, and venules will then merge into veins, which are larger vessels that are going back towards the heart, and then that's kind of our loop. So like we said, blood comes out of the aorta, comes down the descending aorta, goes over into a renal artery. The renal artery is going to get smaller in diameter, and it's going to turn into arterioles. So here is the afferent arteriole, which basically means afferent means going towards. Then we have the efferent arteriole, which is going away from the nephron. So the afferent arteriole, is going to form this ball of capillaries called the glomerulus. And the glomerulus is just a capillary bed that feeds this one nephron. And then there's a vessel that comes out of that capillary bed, which is the efferent arteriole. So afferent goes in, glomerulus is bed of capillaries, efferent comes out. So A before E. And then whatever is in the capillaries of glomerulus is going to flow out of the blood system and then go into the kidney. And that's going to form Bowman's capsule right here. So if stuff goes out of the blood into the kidney, it's going from the glomerulus into Bowman's capsule. So now we're in the kidney. As we go around Bowman's capsule, the first tube section that we're in is called the proximal convoluted tubule. You can also call this the PCT. What happens in the proximal convoluted tubule basically is that we reabsorb stuff that we need back into the blood. And then you can see blood vessels back here. So anything that gets pulled out of the PCT is going to end up back in these blood vessels. And these blood vessels that surround the nephron are called peritubular capillaries. So basically the way that the kidney works is it's cleaning out the blood. So here's your blood. And what basically happens is that we dump everything that we can into Bowman's capsule. And then as we're moving along the proximal convoluted tubule, we're like, oh, we dumped all this stuff out. We actually need that stuff. So sugar and salt and potassium and chloride, we're like, oh, we actually need those things. So then we take them out of the PCT and put them back into the peritubular capillaries. Okay, we'll talk about that more. And then as you go along the proximal convoluted tubule, it's called proximal because proximal means close to Bowman's capsule, convoluted because it's kind of like windy, and tubule because it's a tube. Then we go down the descending limb of the loop of Henle, and then go up the ascending limb of the loop of Henle. So again, descending because it's coming down, ascending because it's going up, and it's called the loop of Henle because it's a loop, and some guy named Henle probably discovered it. So the loop of Henle is unique in that the descending side allows water to pass. The ascending side allows salt to pass. So what that's going to do is basically the outside of the kidney, like we said, is very salty. The cortex and medulla are very salty. So there's a lot of salt over here. Water is going to follow. So it pulls water out of the loop of Henle and into the peritubular capillaries, which are out here. This vasa recta is the blood vessels that kind of surround the loop of Henle. So it's kind of like peritubular capillaries, but we specifically call it the vasa recta because its job is to surround the loop of Henle. So like we said, water is going to come out because the area is salty, and it's going to go back into your capillaries, so now the water is back in your blood system. On the other side, we're going to pull salt out, and when salt gets pulled out, it's going to keep this area very salty. So we can't just keep having water come out to dilute this area without also having salt come out to concentrate the area. So the descending side of the loop of Henle pulls water out so it can go back into your body, and the ascending side of the loop of Henle pulls salt out, which will make this area salty, which on the other side will make water come out. And basically, the loop of Henle, what it's doing is it's causing us to make very concentrated urine. Again, water is hard to come by for most organisms, so you don't want to waste water. You don't want to just pee out water when you need to keep that water inside of your body to stay hydrated. So the loop of Henle is going to help to do that. Some organisms, like organisms that live in the desert, they have evolved to have very, very, very long loop of Henles, so they can make very, very, very concentrated urine. If you have a dog, you'll notice that your dogs make very concentrated urine because, again, water is hard to come by. We don't want to just pee out really dilute water. So what's going to allow that to happen is this loop of Henle inside of all the nephrons, which are inside of all your kidneys. Okay, so the ascending limb of loop of Henle merges into the DCT, which is the distal convoluted tubule. Distal means far away from the Bowman's capsule. So again, it's a windy tube, and what's happening in the distal convoluted tubule is some secretion, so any more stuff in the paratubular capillaries that we need to get rid of is going to get dumped into the DCT, and then some reabsorption. Anything that we didn't pull back into the blood that's still just hanging out in your kidneys can also get pulled back into the paratubular capillaries. Okay, and then we're kind of at the very last stop of the nephron. If you're still in the nephron tubule, you're pretty much going to exit the body pretty soon. So the last part of the nephron is called the collecting duct. And then these branches are just all the other places that the other nephrons are connecting together. So nephrons share collecting ducts, and then the collecting ducts are going to merge together into the pelvis, and the pelvis leads to the ureter leads to the bladder leads to the urethra, which goes out of your body. If you're still in this tubule at this point, you're pretty much going to become part of the urine. The last thing that the collecting duct can do is pull back more water into your body and then not make that a part of the urine. The collecting duct is where ADH works, which is antidiuretic hormone, which is made by your pituitary. ADH basically puts little aquaporins or water channels into the collecting duct and pulls water back into your body if you need to save even more water than you did through the loop of Henle. But then if you have enough water, you don't make ADH, which means there are no little water channels, and then the water just stays in the urine, and then you end up making dilute urine because you probably have so much water that you don't need ADH. So that's what the collecting duct does. And that's kind of like the overview of the nephron. We're going to go through and we're going to look at each individual part of the nephron and see what's happening. So Bowman's capsule and glomerulus, we'll look at that. What's happening with the PCT, we'll look at that. What's happening in the loop of Henle, we'll look at that. And then what's happening in the collecting duct, we'll look at that. The distal convoluted tubule is not that interesting, so we'll kind of skip over that. But like we said, there's going to be some reabsorption and some secretion that occurs. So the basic steps of the nephron is going to be ultrafiltration. So the glomerulus ultrafiltrates. We'll also have selective reabsorption, selective secretion, and then we just end up doing water reabsorption and then excretion, getting stuff out of the kidney and into your bladder. Okay, so like we said, the afferent arteriole branches off of the renal artery, and each afferent arteriole is going to feed one nephron, and it's going to form a ball of capillaries called the glomerulus. The glomerulus is surrounded by Bowman's capsule, which is the beginning of the kidney nephron. So this red part is your blood system, this yellow part is a part of your kidney system. And then the efferent arteriole is going to exit the glomerulus or Bowman's capsule and then return that blood back to your circulation. Okay, so if we look at the capillaries, we know that capillaries are one cell thick, which makes it very easy for stuff to come out, and this is where exchange happens in capillary beds. The glomerulus is also under a lot of pressure, and things like to go from high pressure to low pressure. So if there's a lot of high pressure in here, it squeezes things out into Bowman's capsule. Um, the other important thing to know is that Bowman's capsule is going to be formed by these cells that you can kind of see that surround the capillaries, and only things that are small enough to squeeze between the capillary cells and squeeze between the Bowman capsule cells can actually end up in the kidneys out here. Those cells are called podocytes, which kind of mean like feet cells, but they basically form around the glomerulus, and you have to pass through a capillary cell and also pass by a podocyte to end up in Bowman's capsule. They're calling it a fenestrated wall, which basically means fenestrations are windows. So if you can fit between the two capillary cells, you're fitting through that window, which allows you to hop out of the blood and then into the kidney tubule or the nephron or the Bowman's capsule, whatever you want to call it. So the blood system has a little window that stuff can hop out of, and the kidneys have a little window that stuff can hop through as well. So you have to be able to hop through the capillary windows and hop through the Bowman's capsule windows to end up in the kidney over here. Here's another kind of picture of it. You can kind of see like there's little gaps in between the capillary cells, which allows stuff to go through. And then these are the podocytes, which wrap around the glomerulus, and stuff has to fit in between them too. So in this case, here we are in the glomerulus, in the capillary. This is a red blood cell, kind of side profile. It is too big to fit between the little holes of the capillary, and it is also too big to pass through the holes formed by the podocytes. So therefore, blood comes through the afferent arteriole, stays in your glomerulus, and comes out the efferent arteriole. Blood does not end up going through here, which you can probably understand because you should not have blood in your urine. But what does come out is sodium, which is only one atom. Glucose, which is like 24 atoms, but still smaller than a whole cell. So proteins and red blood cells are too big. They stay in the blood system. Salts come out, water comes out, urea comes out, chlorine comes out, glucose comes out into the kidney. And because they are small enough to slide between the capillary cells and small enough to slide between the podocyte cells, that allows them to make their way into the Bowman's capsule. So ultrafiltration is just filtering stuff from your blood by size. So anything that's small enough to fit past these little windows is allowed to go into Bowman's capsule. Anything too big, red blood cells and proteins, they stay in the blood. Okay? So that's ultrafiltration, and that's what the glomerulus and Bowman's capsule do together. So once you are in Bowman's capsule, you're going to make your way along the tube, and you're going to go to the proximal convoluted tubule. And what the proximal convoluted tubule does is active reabsorption. So about 80% of the stuff that came in through Bowman's capsule is going to get pulled back into the blood by the PCT. So like we said, sodium ions, they're going to be pumped actively from the filtrate, which is the stuff that's in your kidneys, to the space outside of the tubules, and then they're going to pass into the peritubular capillaries, which are the blood vessels that surround the PCT. And the pump proteins are going to be inside of the PCT. So imagine this is the tubule. This is where the salt is, the chlorine is, the glucose, the water, the urea, etc. This is basically your blood, but it's been filtered, so there's no blood cells, there's no proteins. And this is the PCT wall. You can see that it has a lot of these blue cylinder guys, or oval guys. Those are the mitochondria. So we need mitochondria because we are actively transporting stuff from here into your body. So the mitochondria is going to give us ATP to move stuff from here out to here, which is going to be back into your blood vessels out here. So what you want to know is that PCT cells have a lot of mitochondria because they're pumping stuff from the lumen back into your body. You can also see that they have lots of microvilli. We usually think about microvilli as being part of the digestive system, but your kidneys also have microvilli because we need a high surface area to volume ratio to be able to reabsorb all the stuff that we want to from the stuff that is inside of the kidneys. Okay, chloride ions, they are going to go out of the tubule as well, because if you pump sodium from here to out here, sodium is a positive ion, chloride is a negative ion, so chloride will follow. Glucose is going to be transported out from the filtrate as well. Because the sodium is being pumped out, glucose has a system, it's called co-transported. So if sodium pumps out, glucose also kind of hops with it, and they get transported together. So we're going from low sodium to high sodium, so we're pumping sodium against the concentration gradient, but then we are also going to move glucose along with it. And then water. Water is just going to follow because of the concentration gradient that we're forming. So water just moves through osmosis. Sodium is actively pumped using ATP. Chlorine follows because chlorine is attracted to sodium, and glucose gets pumped with sodium. So whenever sodium gets moved from here to here, glucose is going to be co-transported. So it's basically a protein that has two seats, one seat from sodium and one seat for glucose, and you need both of them sitting in the car before you move them out. And then water is going to follow because we just moved some sodium, chloride, and glucose out here, and water is going to go this way as well because of osmosis. But anyway, the PCT is going to reabsorb a lot of the nutrients. So remember, we dumped a bunch of stuff that could fit into Bowman's capsule through ultrafiltration, and now we want to steal that stuff back because we actually want to keep it. The things that we didn't steal back, urea. Urea is still sitting here. Okay. Then we make our way along the tubule and we go to the loop of Henle. And we're going to go down the descending loop of Henle where we allow water to go out. So the loop of Henle, descending side, is permeable to water, which means water can exit. And then the ascending side is permeable to salt, which means salt can exit. And again, like we said, the salt exiting on the ascending side is going to allow the kidney to be very salty, which is then going to draw the water out through osmosis. So what we can see is these different numbers in the middle, 300, 600, 900, 1200, and those are just concentrations. It's getting saltier and saltier. 300 is kind of our base osmolarity, and then we can see that it gets even saltier to 600, even more solute, saltier to 900, even more solute, and then down to 1200, even more solute. And where that's coming from is that we're pumping sodium, or I guess allowing sodium to exit the ascending loop of Henle to cause these different concentration gradients. And then, because of these concentration gradients, water is going to come out because water is going to be attracted to those salty areas, and that's going to cause the urine to get more concentrated as it goes down the loop of Henle, which is why it's changing in color from like a light yellow, dilute urine, to a dark yellow, which is concentrated urine. It's really, it's kind of decent to understand through this picture, but there is a video that I put on Classroom that shows everything kind of moving into the tube and out of the tube, and the numbers around changing. So I think that's a really good video to watch, and I definitely recommend you guys watch it because we can't really see how the numbers are changing as things are moving through the kidney through this picture. So that's definitely a better thing to look at. But I'll try to use this picture to explain it for now. So when the filtrate comes in, it has 300 as the osmolarity, which is equal to the outside. So we don't have any net gain or net loss of water. But as the filtrate moves down the descending loop of Henle, it's getting saltier now. It's 300 in here, but let's say it's like 450 or 500 out here. That's going to cause water to go out because if it's 300 here, this is hypotonic and this is hypertonic. So water is going to come out. As water comes out, that makes the filtrate more concentrated. So now the filtrate maybe is equal to the outside. Now it's 500 and 500. So there's no net movement. But then as the 500 moves down, and now we're in the 600 area, water is going to come out because it's attracted to the 600 area. And as water moves out, now they're both 600. Okay, as the water in the filtrate starts to move down some more, this is 600, but out here is like 750, and that's going to cause it to come out. And then now this is 750, this area is now 900. That's going to draw more water out. So now they're both 900. And now this is coming down, and maybe out here is like 1,000. So then it's going to pull more out. And now they're both 1,200. And then for us, 1,200 is the best that our kidneys can do. So that's as salty as it gets. So now the filtrate inside of the kidney is 1,200. And then we're going to draw out some salt. So when you pull out salt, not water, you're pulling out salt, it goes from 1,200 to 1,000, which makes this area nice and salty. As you move up, you pull out more salt, makes this area nice and salty. So the numbers here are changing, not because we're losing water or gaining water, but because we're pulling out that salt to maintain the salinity of our kidney, the saltiness of our kidney. So then when we come back up, we make our way over to the DCT, which we said doesn't really matter for our purposes. We might pull back some stuff. We might secrete some stuff from the paratubular capillaries back into the distal convoluted tubule, or pull stuff out of the distal convoluted tubule and put them into the paratubular capillaries. Just depends on what's going on at that time. But finally, we make our way over to the collecting duct, and which is the last place that you can exit the kidney and go back into the blood system. So if there's low ADH or low antidiuretic hormone, then you just end up kind of peeing out dilute urine because you're not pulling back the water. If there's high ADH, which means there's high amount of antidiuretic hormone will put little water channels along the collecting duct, and it'll pull water out of the collecting duct, which makes it very concentrated, makes the filtrate very concentrated, so then you end up peeing concentrated urine instead of dilute urine. So this is a good picture to understand. If there's low ADH, there's low antidiuretic hormone, there's no channels inside of the collecting duct, so water just stays in the collecting duct and goes out of your body. But if there is high ADH, we're going to have little pores. Let me see if I can draw them. We'll have little pores for water to exit, and they will look like that, and that, and that. Those are little aquaporins for water, and as the water comes out, it makes the urine more concentrated, but you're saving water. If it's low ADH, there are no channels, so the water has to stay in and go out. Whereas in this case, it does not have to do that. Okay. Last slide is your kidneys obviously are very important to filtering your blood, getting the urea out of your blood system. So if you don't get the urea out of your blood system, if you're not filtering it, then urea is staying in your blood. And like we said, urea is less toxic than ammonia, but you can't just keep it in your blood circulating forever. We have to get rid of it. And so sometimes people have kidney failure. Their kidneys don't work for whatever reason. It can happen through infection. It can also happen if you have heart problems. Because your heart's not working, then the kidneys aren't getting enough blood, and the kidneys get mad when they don't get enough blood, and they're basically being starved for blood, and they start to fail. You might require something called dialysis, which is a machine that filters your blood because your kidneys are not able to filter your blood. So basically what happens is people who have dialysis, they have to go to a dialysis center, and they get hooked up to a machine where their blood is drawn out of their body into this tube, and then it's run through a system where there's filtrate out here, so basically fluid out here, clean fluid, and then it kind of works the same way. There's clean fluid out here, and we pull things out of your blood, So we're trying to pull basically the urea out of your blood. And once the clean fluid out here builds up with urea, it kind of dumps into this container out here. And then we put new clean fluid into here. So let me explain that again. You have blood coming out of your arteries, veins, into a tube. The tube is going to be, I'm trying to think about the word. The tube is going to have tiny little holes into it, semi-permeable. The tube is going to be semi-permeable, which is going to allow small things to come out of the blood and then into the filtrate out here. So urea will get pulled out, and as urea builds up out here, it's going to empty into this. So this is almost like urine. And then we'll have new, fresh, clean fluid get pulled in through this container over here, which is then going to allow more urea to get pulled out of your blood. If you didn't have this guy, then urea would get pulled out at a pretty fast rate at first, but then as the urea is building up, it would come out slower and slower because there's already so much urea out here. So we have to empty the dirty dialysis fluid into here, and we have to pump in new clean dialysis fluid to keep the concentration gradient going. So it's clean out here and it's quote unquote dirty in here in your blood. So we want to keep pulling out the dirty stuff. It's almost like if you are doing laundry. You don't want to just rinse your clothes in water and then leave that water. You want to empty that dirty water and put in new water so you can continue kind of rinsing out your clothes. So basically dialysis is like rinsing out your blood because your kidneys are not able to do it for you. You go up to this machine, you get hooked up to this machine where it draws the blood out and then pulls the dirty or urea waste out of your blood, gets dumped into the used dialysis fluid container, and then nice fresh dialysis fluid will be pumped in so you can continue doing this kind of cycle here. And then the blood is going to return back into your blood vessels and go back into your body. And dialysis usually takes like 3 or 4 hours because your kidneys would normally do this all day, every day, but if they're not working, then you want to kind of filter your blood a couple of times before you return to your daily life. And just a fun fact, now more people are doing this thing called peritoneal dialysis, which basically means they have a tube in their abdomen and they put dialysis fluid into their abdomen. So maybe they put like a liter of fluid into their abdominal area, and that fluid will just kind of draw stuff out, draw waste out of your body, and then at the end of the day, I think it's like 8 hours or something like that, you empty that fluid out of your abdomen, and then that was your dialysis. So it's basically like putting a bunch of clean water into your abdomen, sloshing it around. It collects waste, and then you empty that waste. Hopefully that's like an image you guys can form in your head. I don't think I explained that very well. Okay, what you guys want to focus on is understanding osmoregulators, osmoconformers. You kind of want to know how Malpighian's tubules work, just that they exist and generally how they work. And then what's most important is to understand the nephron and that the glomerulus and the Bowman's capsule are doing ultrafiltration. The PCT and the peritubular capillaries are doing selective reabsorption. The loop of Henle is concentrating your urine. And then the collecting duct is reabsorbing water. And together, the kidney parts are going to be able to filter and regulate the concentration of your urine. So they're going to regulate osmolarity of our bodies and then concentrate our urine and excrete our waste. And hopefully that helps to clear some questions up that you had about the kidney if you already looked at this lecture before I recorded anything. And you hopefully now are ready to go ahead and do the summarizer for the kidney. Okay, hope you guys are having a good week, and I will talk to you soon.